Okay, so you guys on Zoom should be able to see the worksheet here. So this is sort of just a review of the elements and the process of transcription, which we went over on Friday. Kind of just some of the main take-home points. Obviously, there's more detail that you'll want to know than just this, but this is some of the most important information from transcription. So remember, with transcription, we're taking the DNA molecule and we're turning it, or we're making an mRNA molecule from it. So we're getting a message from the DNA that will eventually be made into a protein, but we're kind of in the mid step here. Okay, so what did you guys put for the RNA nitrogenous bases?
Cytosine, thymine, the G word, and
guanine and
adenine.
Yeah, right. So you got 3 of them. So the tricky part with RNA is that there's no thymine. So the T is gone and there's uracil. So you switch out thymine with uracil. I'm really glad you actually said that and got it wrong. So that is really important. There's no T on the RNA side. Now we have a U. So it would be, for you guys on Zoom if you didn't hear that, adenine, guanine, cytosine, and uracil. So A, G, C, U. Those are our nitrogenous bases in RNA. And then, so the base pairing rules with that, I just mean, what does A pair with, what does C pair with, etc. So if we have an adenine, we'll start with the DNA. So we have an adenine, an A on the DNA. What is that going to pair with on the RNA? Uracil, yep. So the A is going to pair with a U. That's the weird one with RNA compared to DNA. And then, so if we have, let's see, we had A. If we have a G, what is that going to pair with? Cytosine, yep. So G and C. And then that reverses too. So C, if there were a C on the DNA, it would be a G that it would pair with, right? If we have a T on the DNA, because we still have T on the DNA, what does it pair with on the RNA? Adenine, yeah. So T pairs with A. So there's still T's involved, but it's only on the DNA side of things. If there is a T, it pairs with an A, just like you guys learned before. The only difference there is if there's an A on the DNA, now you have a U on the RNA. Tricky and a little bit, it's easy to get confused there. Transcription requires one main enzyme, wonderfully, or protein enzyme, however you want to think of it. What is this called? RNA polymerase. Yep, RNA polymerase, just like DNA polymerase was kind of the workhorse in DNA replication. RNA polymerase essentially does everything for our intents and purposes in transcription. Okay, which 2 DNA bases, so of those 4 letters we talked about, which 2 DNA bases signal the starting point for transcription? So there's a bunch of them in a row at the very beginning of the DNA strand. Adenine and thymine, thank you. So A's and T's. So if we're looking at the DNA molecule here, so there are still T's. If there's a bunch of A's and T's, that signals to RNA polymerase, hey, bind here. This is where, right after this is where we're going to start transcription. A's and T's, okay. 3 main steps of transcription. What's like the big overall steps? What's the first one? It has to get started. Initiation. And then number 2, elongation. And then at the end, termination. Yeah, so they all kind of make sense. Those are the 3 main steps. We saw that in DNA replication, and we'll see that in translation. So all of these processes that involve basically matching up the genetic code, it's initiation, elongation, termination. The sub-steps within each of those differ, but the main steps remain the same. All right. So we don't have to go through this whole string. We'll just look at each unique letter, and based on the base pairing rules we talked about, I just wanted you guys to build. So I just showed you one half of the DNA here, right? So this is one strand in the double helix. We'll imagine it got unzipped, and this is what we want to make the messenger RNA molecule off of. So here we have the 3' end. So what end of the mRNA is this going to be if we're building it in this direction? 3' matches up with what? 5'. Yeah. So if you guys on Zoom, the 3' end of DNA, the mRNA, that end will be the 5'. It's still anti-parallel. I don't know. Anti-parallel. So 3' matches up with 5'. If we have an A on the DNA, what's going to be put in on the mRNA? U. Yep. So here we start out with the new one, right? A is going to pair with U. If we have a thymine, a T, what's that going to pair with? Still the same as with DNA, yeah, an A. So T and A, so we have another T. A G is going to pair with, what's going to come in here? C, right? So for you guys on Zoom, we have a U will be down here. With the T, there will be an A, a couple A's in a row. With the G, on the other end, on the mRNA, there will be a C. And then if there's a C on the DNA, what pairs with that? A G. Yeah, so you can keep building it. I think we covered all of them. Yeah. And we'll end up with 3 U's at the end because we have these 3 A's. So it'll be 3 U's. And then this will be the 3' end of the mRNA because it's the 5' end of the DNA. Any questions about that worksheet? Okay. Use that when you're studying for, those are some, like I said, some main concepts. So your next quiz will be Friday, and your next exam, I can't remember off the top of my head when it is, but for your next exam, you can use this. I think it's the last day of class, right? Yeah, the very last day of class. So you can use this as kind of a study guide as well. Okay. So we're good with that. Stop sharing, and then so we've done transcription. Seems like you guys have a pretty good understanding of it, which is awesome. It helps having already gone through DNA replication. All right, so let's move on to translation. Really the last part of this kind of molecular view of the whole DNA to protein process. All right. There we go. Okay. Translation of the genetic code. We obviously just finished with transcription. We just talked about it. So as a reminder, with transcription, like I said at the beginning, we're going from DNA and we're making an RNA molecule, specifically an mRNA, which is messenger RNA. We'll talk about 2 other kinds of RNA today that are involved in translation. So we go from DNA to RNA with transcription. Now we're on to translation. We know what's happening there. We're going to take that RNA molecule we made in transcription and we're going to use it to build the eventual protein. So this is the very end of the central dogma of biology, DNA to RNA to protein. We're doing that last step, RNA to protein. So here's a nice diagram, kind of a nice simple diagram thinking about transcription and translation in relation to each other. So here we have our double helix of DNA. It gets unzipped, and then we're making this RNA molecule, which is this pinkish red color, based off of one of those strands, just by the simple base pairing like you guys just did on that transcription worksheet. We have that mRNA molecule. It's called a transcript, RNA transcript. That is going to be read. That's what we're going to talk about today, how these letters are essentially read to put together a bunch of amino acids. And amino acids are the building blocks for a protein. So if you string together a bunch of amino acids, you get the protein. that we're eventually is kind of our goal of this whole process. So we're going to learn how this code is read today and the process of how it's converted into a string of amino acids, also called a polypeptide. So polypeptide, that term will come up again. It just means a string of amino acids. Something a little bit different happens when we're going from that mRNA molecule to the polypeptide. Instead of reading each letter individually like we've kind of been doing, now that code on the mRNA is going to be read in groups of 3. And this is called a codon. So a sequence of 3 of these nitrogenous bases, those letters we were just talking about, will be read at a time. And those 3 will code for 1 amino acid. So this is called a triplet code. One of these would be a codon. Overall, we call this the triplet code for creating polypeptides. Each of these 3-letter combinations, so AUG, codes for a very specific amino acid. So AUG is one that only codes for what we call methionine. Amino acids are, I think throughout this lecture, and often are abbreviated with 3 letters. So you guys don't really need to know the names of the amino acids. I'm not too worried about it. If I ask you about them, I'll just use these abbreviations. But AUG is always going to code for methionine. Most of the others, there's some overlap. So we don't have any overlap here, but different groups of those 3 will sometimes code for the same amino acid. And I'll show you what that looks like. We have a whole chart of the possibilities of these combinations of 3 nitrogenous bases and exactly what amino acid they code for. That's the genetic code. So I'll talk about that. But the process is just that's involved in just reading these nitrogenous bases 3 at a time. That will bring in a specific amino acid, and then all of those will be strung together into the polypeptide. So that's translation in a nutshell, yes.
What does the triplet code code for?
The triplet code is just using 3 letters at a time to create the polypeptide chain. Yeah. So one codon within the triplet code would be, say, AUG, just a group of 3. The triplet code overall is kind of what we call this process of using 3 letters to code for 1 amino acid. Good question. Okay. Where's translation happening? So if you remember, we were talking about transcription has to happen in the nucleus. We're going to ignore prokaryotes. I don't really want to talk about prokaryotes, so we're just going to ignore them. We're talking about eukaryotes, which have a nucleus. Prokaryotes do not. So eukaryotes, transcription happens in the nucleus because that's where the DNA is. So the DNA has to be read in the nucleus. That mRNA that's created then makes its way out of the nucleus, and translation just happens out in the cytoplasm. In the cytoplasm, you have these ribosomes, which you may or may not remember these organelles from a very long time ago when we talked about the organelles of a cell. But ribosomes are going to come back in force today when we're talking about translation. So this mRNA will end up at a ribosome. That's where the whole process of translation takes place, but just out in the cytoplasm. We're not in the nucleus anymore. I'll cover this kind of similarly to how we covered transcription. There's a few additional pieces that I'll have to define, but kind of just go through the overview, what tools are needed, and then the whole kind of the details of the process. Okay, so the basics of translation, we already talked about this. We're taking an mRNA molecule and we're making a polypeptide chain, which is just a chain of amino acids. The code that is used on the mRNA is called a codon. So those three groups of three, those are all codons. That's what's read. Interestingly, that code is universal among all eukaryotes. And what I mean by that is AUG, that specific codon, is always going to code for the same amino acid no matter what eukaryote you're looking at. A human, a dog, a jellyfish. It's the same across all eukaryotes, which is pretty amazing. So the genetic code is universal. And then we still have complementary base pairing. So you guys are familiar with that idea where you're matching up letters that complement each other, right? Like we just did. The A, the T, the A, the U, the C, and the G. But now we're going between that mRNA that we made with transcription and a whole new kind of RNA, which is called tRNA or transfer RNA. I'll define these, well, I'll define the three different kinds of RNA on the next slide. We have new RNA types coming in, but still the same base pairing rules. It's still RNA, so we still have a U instead of the T. But the base pairing rules are the same. Here's our three types of RNA. You guys are already familiar with the messenger RNA. This is what gets made in transcription. Sorry if I'm repeating myself a lot, but hopefully it's kind of helpful to hear this over and over again. mRNA is what gets made in transcription, makes its way out of the nucleus, and takes that message from the DNA. So that's why it's called messenger RNA. It takes a message from the DNA to create a protein, so out of the nucleus to the ribosome. Somehow we have to get the amino acid from out in the cytoplasm or wherever it is to this ribosome so we can string a bunch of them together. The molecule that does that is called tRNA or transfer RNA. So it's transferring the amino acids from the cytoplasm to the growing polypeptide chain. So it's bringing in, and I'll show you kind of what that looks like in a simplified diagram form, but it's bringing in an amino acid, one amino acid at a time, to create this long chain of amino acids. Our final kind of RNA, I'm not going to talk about really in much detail other than it's rRNA. It's what makes up the ribosomes. You don't need to know anything more about it than that, other than it's sort of the building block for the ribosomes, which is where this translation process is happening. So that is ribosomal RNA. It's interesting that DNA gets all the attention, basically. So most people, even if they haven't taken a biology class, have heard of DNA. A lot of people haven't heard of RNA, but RNA is kind of the workhorse for this whole process. DNA stores the info, which is very important. RNA is doing all the rest. It's doing the rest of the work. It's taking the message, it's bringing in amino acids, it's making ribosomes. So it's a really, really important molecule. All right, some tools that we need. Not a whole lot. I kind of mentioned them all, actually. We need amino acids. These are attached to those tRNA molecules. So we got to bring in the amino acids, which involves the tRNA. I'm not going to talk about the process of how these are attached, the charging process. But when an amino acid is attached to a tRNA molecule, it's called, the tRNA is referred to as charged. So it's charged when an amino acid is attached to it. We have our ribosome, which is ribosomal RNA, or rRNA plus proteins to make it. We have our mRNA transcript that we made in transcription, and then we need some energy. I won't talk too much about where the energy is used, but just know energy is needed for this process. Okay. Yeah, I'll go over this briefly. Let's ignore the prokaryotic again. I mean, not that it doesn't really matter. We can look at prokaryotic or eukaryotic here. Eukaryotic is just more complicated. But the take home here, translation of an mRNA, the protein coding segment is the only part that's actually read by the ribosome in order to make the protein, obviously. So remember, we have this coding segment. All the rest is kind of mostly for protective purposes and also kind of a guide for where to start and stop the process. But only the regions between the start and stop codons, which I'll talk about those in a minute. Those are sequences of those three nitrogenous bases that code for, well, start codon will code for a specific amino acid. The stop codon, which is at the end, so there's a few different stop codons. So 3 different ways to indicate to this process to stop. Those don't actually code for an amino acid, they just tell the process to stop. I'll talk about that more in a minute, but the protein coding segment for our purposes starts with a start codon and ends with a stop codon. Right, so here's the tRNA, which is really, it looks really strange. It doesn't look like the rest of the RNA that we looked at. These are just 3 different views of it. I'll go over what they are in a second. View C, you can see there's still just it's just a chain of these nitrogenous bases, the A's, the C's, the G's, and the U's. So same idea, it's just folded really strangely. So rather than it being a nice straight molecule like the mRNA, it folds over on itself and looks really complicated. But it's really just a string of nitrogenous bases still. This one in the middle, I should probably block out, but it's the 3-dimensional structure. It's, yeah, you don't have to worry about that too much. You might see this shape, kind of like a, I don't know what you would call that, but that weird shape in your book. I think this is the shape they use mostly in your book, if you have a book, but you'll see throughout the rest of this presentation, tRNA will, I think for the most part, be, this will be the diagram for our transfer RNA molecules. We don't really care about most of these nitrogenous bases. We care about the 3 at the bottom, which are going to show up here on this diagram. So here's our transfer RNA molecule for our purposes. At the top is where the amino acid will attach. So we don't have any amino acid attached here yet. So this is not charged. The complementary base pairing comes in down here with the codon and then the anticodon. So if we're imagining down here, we have the long mRNA molecule, we'll say on the bottom. Oops. So for you guys on Zoom, this would be the mRNA molecule. We're just looking at 3 nitrogenous bases here, UUC. Just one small portion of that mRNA strand. The tRNA that comes in will have the complementary bases to that UUC. So what we call that on the tRNA is the anticodon. So you have the codon on the mRNA, the anticodon on the tRNA. They're complementary bases. We have a U, so we have a U on the mRNA, so we have an A on the tRNA. Another U and A. We have a C at the end of this codon in the mRNA. So on the anticodon, you get a G. Same base pairing rules, like I said. And it's done 3 at a time. Each of these tRNAs has 3 letters for each anticodon. And those are the only 3 letters we really care about in this whole tRNA molecule. Like I said, there's a bunch of others we can see, but we don't care about any other than these bottom 3. Okay, I think that's the main, important parts of the tRNA. Now let's look at the ribosome and kind of how all this fits together. So this brown thing here is the ribosome. There's a small part and a large part. You don't have to worry too much about that. But overall, the ribosome has these 3 empty sections to accept this tRNA. So here we have our tRNA molecule. And now we have an amino acid actually attached to it. So that's our amino acid that's attached. So this tRNA we would say it's charged. It's charged with an amino acid. So essentially what happens is, you can think of it 2 ways, I guess. The ribosome, you can think of the mRNA as being stationary, and the ribosome moves along the mRNA, or you can I kind of think of it this way, the mRNA is moving through the ribosome and kind of being pulled along, exposing 3 of these nitrogenous bases at a time. So 1 codon at a time. You don't have to worry about there's 3 sites here, the E, the P, and the A. Don't worry about the names of those or what they're for. I'll talk a little bit about them, but you don't have to really know much information about them. But there are these 3 individual sites on the ribosome that will accept tRNA molecules. The tRNA molecule that comes in, again, has to match up with this codon on the mRNA. Okay. So that is a ribosome. Okay. So now we have the genetic code. Like I said, this is universal among all eukaryotes. So you can find a codon in here, and it will apply to us. It will apply to your cat or your dog or your turtle, which is pretty amazing. So this is essentially showing what those 3-letter combinations, the codons, which amino acid is going to be brought in. So for example, here we have the first position options over on the left there. So for you guys on Zoom, here's the first position. So let's say we have a CCA. We'll say that's our codon. So first position will be C. The second position you find across the top. Here we have a C, so C and C. And then the third position is over here. So you find the first, second, third letters, and you figure out where they intersect. So here we have A. So we'll go over to the CCA, and you can see it here, CCA. That's going to code for pro, which is short for proline, so a specific amino acid. And you can see there's overlap. So CCA codes for that. 3 C's will code for that, CCU. So there's overlap in this. There's what we call redundancy. There's a few that only code for one thing, and I'll talk about those on the next slide. You can see those here. AUG, UAA, UAG, UGA, and then UGG. So I'll talk about the AUG and then these stop ones on the next slide. But that's generally, it's really easy to read. You find the first letter, you find the second letter, you find the third letter, figure out where all those intersect, and you can figure out what amino acid is going to be coded for. It's really important to remember that these 3-letter codons that we're talking about when we're reading this is from the mRNA. We're not talking about the tRNA, which has the anticodon. This code is on the mRNA. So yeah, like I said, the amino acids, I'll pretty much always have abbreviated by 3 letters. If I ask you to find out what amino acid is going to be coded for on an exam, I would give you this genetic code and then let you figure out what the codon codes for. So be able to read this. I guess that's basically what I'm saying there. I might ask you, what does ACG code for, or something like that. A few of particular importance, like I just mentioned. One, you just want to memorize. So I might ask you this without giving you the genetic code, is the start codon. That is AUG. So if you find AUG, it's right here. It always codes for met or methionine. In every single polypeptide, this is the very first amino acid. This signals the start of translation. So an easy way to remember AUG is the start codon is that school starts in August. So AUG, that's how I've remembered it since high school, I think. So that codes for methionine. You want to know that it codes for methionine, and you want to know the AUG, that it's the start codon. There's 3 stop codons. I won't ask you to memorize them. I'll give you the genetic code if I want you to find out what these codons code for. But they're UAA, UAG, and UGA. So they're all right here. And you can see they just say stop. So they do not code for an amino acid. They don't bring an amino acid in. The start codon does. It brings in methionine. The stop codons just stop the process. They don't bring in any new amino acid. It just stops the process. Everything breaks apart, which we'll talk about, and translation's done. So on Wednesday, we'll get into genetic mutations, and you can imagine if some of these letters are off, even one letter sometimes, it can really mess up a protein that's being built. And even one letter a lot of times can cause a pretty serious disease. So sickle cell anemia we'll talk about, that's caused by one of these letters being off in a specific gene, obviously. But just one letter being off can make a huge difference. Sometimes it doesn't matter at all. Like I said, there's a lot of redundancy. So if a codon was supposed to be CCA, but it ends up being CCC, it's going to code for the same amino acid, so it doesn't really have an effect. We'll talk about that more on Wednesday. I think that's all I want to say about the genetic code. Any questions about this? Okay. Moving on. The mechanics of translation. So what's actually going on? You kind of understand the tools that are needed and sort of that base pairing process. I talked about tRNA charging. Just a reminder here what that means. Before translation can start, or before a tRNA can bring in an amino acid, they have to come together. So that's called charging the tRNA. Once an amino acid is added to this weirdly shaped tRNA molecule, it's charged and it's ready to go. It'll move towards the ribosome when it needs to. So that's kind of I guess a pre-translation process. Oh, yeah, so now more into the mechanics. We have the 3 stages again: initiation, elongation, termination. We have to start the process, we have to make that polypeptide chain grow, and then we have to stop the process. Same idea, just we're making a different thing this time. We're not making DNA, we're not making mRNA. Instead, we're making a polypeptide chain. So here's a simple diagrammatic overview of the process. So I'm just going to sort of describe what's being shown here. And then on the next few slides, I have the bullet points of exactly what's happening in each of these stages of translation. So we'll start up here. Here we have the ribosome, the mRNA, the tRNA with the amino acid attached. So for you guys on Zoom, we have the mRNA transcript, the brown ribosome, the tRNA, and then the amino acid up here. So we already have one in there. It's already matched up, the codon with the anticodon, brought in whatever amino acid it needs to. If this is the beginning, it's that methionine amino acid. Then another tRNA with another amino acid will come in to this second spot. Again, don't worry about the EPA. But these two tRNAs will then be next to each other. Both of them are matched up, codon to anticodon. This tRNA has a very specific amino acid attached to it. And then in order to actually, we need to attach these amino acids because we need to make it a chain, right? So this amino acid, I'm sure there's a complicated process to it, but I just imagine it as kind of jumping over and attaching to this amino acid. So, and that's what, there's a little arrow here that shows it moving over. So from this tRNA, the first amino acid will attach to the second one that just came in. So here we have that elongation of the polypeptide chain. So now this tRNA is empty, it's useless, and it gets kicked out. So it leaves off to go get another amino acid for another translation process. And then this one that was in the A spot shifts over. So we have it shifting over. Now we have two amino acids. That whole process starts over again with the new tRNA and amino acid coming in. And then the polypeptide chain will just grow as more and more come in. So that's a general overview of what's going on. And I think I mentioned this elsewhere, but the bond between the amino acids, it's called a peptide bond. I'm pretty sure I have that listed elsewhere, but you want to make note of that. Peptide bond is this bond between the amino acids. So for you guys on Zoom, it'd be this little bond between these two purple amino acids. That's a peptide bond because we have a polypeptide chain. Okay, so now the word version, the bullet pointed version. The initiation process. So we'll do initiation, elongation, termination. So the ribosome and the mRNA come together. Binding of the ribosome to mRNA. And that start codon is detected. So AUG, remember that's the start codon. So AUG is found on that mRNA, and that's where the translation process will begin. That means the tRNA with the anticodon to that stop codon will come in. It always contains MET, or methionine amino acid. And then the process starts. So energy is needed here, but that's the whole process of initiation. Ribosome binding to the mRNA, and then that first tRNA coming in to signal the start of translation. Okay. So now we're ready to go. Here's an image of what that initiation kind of looks like. Like I said, there's two different pieces to the ribosome. Don't worry too much about that image over there. We don't have the second part of the ribosome yet. Here we have the whole complex together. So you have the ribosome, you've got your mRNA binded to it, and then you've got that first tRNA with the starting amino acid MET. Next, elongation. So you guys probably have a good idea of what's happening here. Now we just bring in tRNA after tRNA with the right amino acid, ideally, based on the codon on the mRNA. Peptide bond formation will occur as that the amino acids get transferred into one long chain. And the ribosome will move along that mRNA from the 5' to 3' end of it, just reading those codons. So 3 letters at a time, they'll be reading those codons and signaling for the correct tRNA to come in and bring with it the correct amino acid. As those amino acids get transferred over and grow into that long chain, the tRNA that is now uncharged, that's lost its amino acid to the growing chain, will get kicked out of the ribosome. It's been used. And then, let's see, yeah, so the whole cycle is repeated for the next codon. We still need energy for this process. So initiation and elongation require energy. Any questions so far? So we're building our amino acid chain. It's getting longer and longer as we bring in one tRNA after another. So what does this look like? I don't know, pictures always help me. Hopefully they help you. So here we have, we're kind of in the middle here. So we already have a polypeptide chain growing. You can see the purple amino acids up here. Oops. And then subsequent tRNAs bring in one amino acid at a time. So we're bringing in one more amino acid. That chain that's growing will then be transferred to the new tRNA that came in, and that amino acid. Everything kind of shifts to the left here within the ribosome, or you can think of it as the ribosome shifting to the right, however you want to think about it. So we've got peptide bond formation occurring between those amino acids. Now this tRNA, poor little guy, he doesn't have an amino acid attached anymore, so he's going to get kicked out. He's not really useful anymore. He gave up his amino acid and will leave the ribosome. So he'll get kicked out. The tRNA with that really, well, eventually long, but growing amino acid chain will shift over to the middle, freeing up that first space for a whole new tRNA to be brought in with the new amino acid. Starting that whole process over again, just growing that chain of amino acids. Good. I don't want to go too fast for people. Nope, there we go. Repeat the process with the next tRNA. So it just happens over and over again until we get to a stop codon. So we'll imagine we got to one of those stop codons. We'll say, I don't know, maybe it was UGA. I don't know what we have on the next slide, but we're at a stop codon. There's 3 different stop codons. We're at one of them. When the stop codon is reached, what's called a release factor will come in. So it doesn't bring in a new amino acid. It just stops the process. And then essentially everything breaks apart, except for the polypeptide chain. It stays together, but breaks off of that, away from the ribosome, off of that final tRNA. So the final tRNA is released. the polypeptide is released and the ribosome disassembles. And this all requires energy as well. So lots of energy needed for translation. Okay, so let's see what this looks like on our diagram here. Here we have our ribosome. We got to one of our stop codons. Here's the 3 options. We'll just say that's UGA, for example. A release factor is brought in. So they make it a different color in this diagram, which is helpful, indicating that there's no amino acid attached to it. So we don't have a new amino acid. It just goes into that 3rd slot in the ribosome and stops the whole process. That last tRNA is released, the polypeptide chain is released, and then everything just breaks apart. So the mRNA goes on its way, the ribosome breaks apart to be used again later for other translations. We're not going to go into this. I think I mentioned it when we talked about proteins, but after this, so we have this chain of amino acids and there's all this folding, right? So proteins fold kind of over on themselves again and again to be functional. So it's not fully functional, but the basics are there. So the chain of amino acids is obviously the starting point to make a protein. So after that, a lot of stuff happens, but we're not going to talk about it because this is the end of translation. All right, I think that's a great stopping point, and we'll actually watch a summary video of the process. Yeah, we'll start with that on Wednesday. So yeah, that's it for now. We've done transcription and translation, and I'll see you guys on Wednesday.